Hello everyone, welcome back to pharmabahen. Today we are starting antimicrobial drugs. Antimicrobials are the drugs that selectively act on the microorganisms, either killing them or inhibiting their growth with minimal harm to the host. This word selectivity is very important for the exams. Why means microbes have different structure, humans have different structure. Drugs target differences, not similarities. This concept is called selective toxicity. The ideal antimicrobial should have maximum selective toxicity. Difference between the chemotherapy and the antimicrobials. So now many students get confused here. So listen carefully. Chemotherapy, treatment of diseases using chemicals. Antimicrobial chemotherapy, the treatment of infection using chemicals that act on the microbes. So in pharmacology, when we say chemotherapeutic agents, we are mainly talking about the antimicrobials. What decides the action of an antimicrobial? So an antimicrobial does not act randomly. It acts depends on the structure of microorganisms, site of infection, drug concentration, host immunity. So that's why some drugs work in one infection but fails in another. So, and also coming to the bactericidal versus the bacteristatic. Before getting into the classification, one more concept must sit in your brain, that is the bactericidal drugs which kills the bacteria directly, useful in severe infections, examples like meningitis and endocarditis. Coming to the bacteriostatic drugs, inhibit the bacterial growth and host immunity, clears infection. So important, bacteriostatic drugs becomes bactericidal at higher doses. And why we have to classify the antimicrobials? Now comes to the most logical question, why so many classifications? Because antimicrobials can be classified based on the spectrum, chemical structure, clinical uses, mechanism of action. Most important one, mechanism of action tells us how drug works, why resistance develop, why side effects occur. So once you understand the mechanism, you don't need to blindly memorize everything or anything. So mainly we have five types of classes, like first one, the inhibition of cell wall synthesis, disruption of the cell membrane, inhibition of the protein synthesis, and the fourth, inhibition of nucleic acid synthesis, and the fifth, anti-metabolites. And the examples for the inhibition of cell wall synthesis is like penicillins, like penicillin G, cephalosporins, carbapenems, and vancomycin that acts on the peptidoglycan synthesis, binds to PPBs and cause bacterial lysis and bactericidal. Coming to the disruption of cell membrane, we have polymyxin B and colistin. So it acts like detergent, increases the membrane permeability, cause leakage of the cell contents. Then coming to the inhibition of protein synthesis, that is 30S ribosomal subunit, aminoglycosides like gentamicin, tetracyclines, doxycycline, 50S ribosomal subunits like macrolides like erythromycin, chloramophenicol, clindamycin. So they act on the 70S bacterial ribosome, mostly bacteriostatic. So ours is 80S, so they won't attack our human cells. And aminoglycosides are the bactericidal. And coming to the inhibition of nucleic acid synthesis, examples like fluoroquinolones, rifampicin, and metronidazole. So they block the DNA gyrase or the DNA polymerase, prevent replication and the transcription. They are the bactericidal. Coming to anti-metabolites like sulfonamides, trimethoprim inhibits the folic acid synthesis. Human gets foliate from the diet, so bacteria synthesize their foliate. So because this helps in the selective toxicity, now we are entering into the most important antimicrobial topic, that is beta-lactam antibiotics. Before we touch the drug names, remember that this should not be memorized. It should be understood first, because beta lactams are not drug based, they are the bacteria structure based. Okay, everything in the beta lactam revolves around one structure, that is a bacterial cell wall. Let us revise it properly. The bacterial cell wall is made up of the peptidoglycan. So the peptidoglycan have the longer sugar chains and a peptide cross links. So think it as like sugar chains or like bricks, and the peptides are like cement. So without the cement, wall will collapse. So the function of the cell wall is like it maintains the shape, protects from the osmotic pressure, essential for the bacterial survival. So human cells do not have cell wall, so perfect site for the selective toxicity. And now here is the exam winning concept. Bacterial cell wall is synthesized by the enzyme called penicillin binding proteins. So penicillin binding proteins are responsible for the cross linking of the peptidoglycan and giving strength to the cell wall. So without PPBs, cell wall will be weak and the cell will burst. Now, finally, what is the beta lactam? So, beta lactam antibiotics are the drugs that contain a beta lactam ring, and it inhibits the bacterial cell wall synthesis by binding to the PPBs. So, there are three keywords: beta lactam ring, PPBs, and the cell wall synthesis inhibition. Let us come to the mechanism of action. Beta lactam drug reaches bacteria, and the beta lactam ring resembles D-alanine and D-alanine. So the drug binds to PPBs and cross-linking of the peptidoglycan stops. So because of this, the cell wall becomes weak, osmotic pressure increases, bacteria cell lysis occurs. So the final result is like bactericidal action. So we are little confused about the cross-linking of peptidoglycan, right? And the structure of the bacteria. So let us go deeper into that. If you understand this structure once, you will never forget the penicillin, cephalosporins, and resistance. So today there is no drugs, only the structure, logic, and clarity. Then after we will moving forward with the classifications part. So what is the peptidoglycan? So the bacterial cell wall is mainly made up of the substance called peptidoglycan. Let us break the word. Peptido means amino acids. Glycon means the sugar chains. So the sugar is a backbone. Peptide is helping in the cross linking. So the two most important sugars are the NAM and the NAG. So NAM means the N-acetyl mumeric acid, and the NAG means N-acetyl glucosamine. So they are arranged like this. Now listen carefully. Nag is just a sugar, but the Nam is a sugar with a tail. So Nam has the peptide side chain attached to it, and the peptide chain usually contains L-alanine, D-alanine, D-glutamic acid, lysine or mesodap. So it has five amino acids. So coming to the main point, the bacterial cell wall has two types of bonding. So there is a trans glycosylation, that is sugar-sugar linking, like NAM and the NAG linking. So they form the glycan chains. This step builds the length of the wall. So now coming to the second one, that is transpeptidation, the peptide-peptide linking. So linking between the peptide chains of the NAM and to the NAM. So it gives the strength and rigidity. Think of it as the cementing the bricks. So the most important lines like transglycolation is for length and the transpeptidation is for the strength. Now here comes the enzyme you must remember for your life, that is penicillin binding proteins. The penicillin binding proteins are the enzymes that carry out the transpeptidation, that is cross-linking of the peptide chains. So this will strengthen the bacterial cell wall. So without the PPBs, there will be no cross-linking, there will be a weak cell wall. So where do beta lactams act? Now everything connects. Beta lactam antibiotics do not affect the NAG, do not affect the NAM formation, do not affect the synthesis of the sugar. They specifically inhibit the transpeptidation. So by binding to the PPBs. So what happens when the transpeptidation stops? So let us follow the sequence slowly. NAM and the NAG chains was formed, but the peptidation cross-linking does not occur. So cell wall becomes weak and bacteria cannot withstand the osmotic pressure. The cell will burst. The final result is the bactericidal effect, as we see. Classification of the beta lactams, and we will move forward with the other antimicrobials in this series. Thank you so much for watching the video, and please do follow if you like the content.